I am really excited to launch our first talk of the day, which is Breath from Salt, a history of cystic fibrosis from characterization to almost cure. We have Ms. Bijal Trivedi, who is an author and senior science editor of National Geographic, joining us today to talk about this topic. And again, I will just note our continuing education, accreditation, and disclosure statement, as well as the CEU tab in the attendee website. Again, I mentioned that none of our speakers at this year's summit have conflicts to disclose. And again, just a reminder that to claim those credits, you will be receiving a survey at the end of today. With that, I'm going to stop sharing my screen and ask Bijal to join us and share hers.
Hi, thank you so much for inviting me here today. I'm Bijal Trivedi, and I know that I'm speaking to a lot of experts, many of whom know a lot more than I do about genetic disease. But what I am hoping to share with you today is why I decided to write a book about a rare disease, cystic fibrosis. And the reason is that this is really an undertold story in the history of medicine. And I think that there are a lot of lessons that other communities can take and use in their fight against genetic diseases. And that's what I would like to share with you today. Can we have the next slide, please? So I should, pardon me, I should tell you that this book took me 10 years to research and write. And that's because it's a really complicated story. Next slide. So my interest in cystic fibrosis began when I was asked to write a story for Discover magazine about a new drug. And the new drug was called Kalydeco. And it was the first drug that ever had been developed to treat cystic fibrosis patients. Next slide. So there are two things that immediately struck me when I began researching this story. And the first was that, cystic fibrosis is a rare and deadly genetic disease. And there are only about 30,000 people in the US who suffer from it, and about 75,000 worldwide. And the second issue was that Kalydeco, which was the first medicine that Vertex made for cystic fibrosis, it only works for 4% of the population. So that's 1,200 people. And that was a tiny market. So I was wondering, why would anybody or, a major pharmaceutical company bother to make a drug for such a small number of people? And as I began interviewing people, I realized that, I'd stumbled on a 70-year epic saga and one of the great triumphs of the new era of genetic medicine. And I wanted to be the one to tell this story because it was fantastic. There was a lot of joy, but, sadly also a lot of grief. There was triumph and there was failure, and there were monumental scientific breakthroughs and heartbreaking setbacks. Next slide. So one of the amazing things about this story is that, I came to this with a very different perspective. And I believed, as Proust says, that I came to the story with what he describes as other eyes. And that means that, I didn't know anyone that had cystic fibrosis. I barely knew anything about the disease. But what I did have was a background in science. So I could appreciate the huge number of breakthroughs and the number of pioneers in this field. And as a journalist, I could also recognize the stories that every patient had a story, every family and physician had a story. And I wanted to combine these. I wanted to weave together the science and the stories of the family and the science of this foundation, the Cystic Fibrosis Foundation, and show how these all came together to guide the development of treatments. Next slide. So initially, I was completely overwhelmed. There were so many elements to this story. But, as the theme of this conference is joining the dots, what it came down to for me was that I had to join the dots. I had to figure out how all these parts that you see in front of you fit together and tell these stories. And it was pretty overwhelming. And so I want to show you how I did this. And I focused on 3 different themes or threads, and I tried to weave them within each other and in and out during the stories. So the first thread was family, and all the families and the patients and how cystic fibrosis had affected their life. The second thread was science, all the scientific breakthroughs that were required to bring us to the point today where we have therapies for 90% of people with cystic fibrosis. And the third thread of this story was venture philanthropy, how the cystic fibrosis used this thing called venture philanthropy to fund the development of drugs for this rare disease. Next slide. So, starting from a 10-page magazine article and going to a book project is a huge step, a huge jump. But the event that really changed my life and made me commit to going beyond a magazine story was meeting Joe and Kathy O'Donnell. And they were the parents of Joey O'Donnell, who suffered from cystic fibrosis, and ultimately he died from the disease after 12 short years. And you can see the family here. And what Joe and Kathy shared with me was the most intimate and moving details of their precious 12 years with Joey. They shared the torturous 6-month period through which Joe and Kathy struggled to get a diagnosis for Joey as he became sicker and sicker. They told me about how he thrived after the diagnosis and about the 5 relatively healthy years he had, and how he grew into a charming young preteen. And they also revealed the rough times when Joey's health began slipping away. So if you look at this Maggie O'Farrell quote, "To raise a child with additional needs is to inhabit a different country from those around you. You will have your own customs, rules, rituals, habits, mores, and vocabulary. People may visit, but they will never truly know what it is to live within that border." And what surprised me most about the O'Donnells is that they took me into this world within that border that Maggie O'Farrell describes. So I'd like to share a chapter for you, with you from the first chapter of my book. And this begins in 1974, when Joey was just 6 months old. 10 days later, on October 20th, Joey was readmitted. Nearly 6 months old now, he still only weighed about 8.5 pounds. He was vomiting again. His body was lethargic and floppy. His skin was covered in an ever denser coat of speckles, and he had a raging case of pink eye. Dr. David Walton, a pediatric ophthalmologist, examined him and found that Joey's eyes were dry, which left the surface vulnerable to infection, and the clear protective covering of the eye, the cornea, was cloudy. Joey was looking at the world through a fogged windshield. Dr. Walton suspected a severe lack of vitamin A, the most common cause of blindness worldwide. But after consulting with Kathy, he found that there was no shortage of vitamin A, rich foods in Joey's diet. There was, however, another possibility, which Walton knew was linked to such eye problems, an inherited disease particularly common in New Englanders of Irish descent. Walton suggested that Joey's sweat should be tested. Kathy told him that this had already been done months earlier and that the results were normal. But Walton knew that the test was often botched by inexperienced doctors at small hospitals. So he reordered that tests be done at Mass General. On the next day, October 21st, 1974, Kathy was standing by an elevator when a young resident who had been caring for Joey stepped out with the results. The sweat test had confirmed Walton's suspicions. Joey had hyper-salty sweat, the resident explained, and it was the hallmark of an inherited and fatal disease called cystic fibrosis. Kathy was terrified. She had never heard of cystic fibrosis and had no idea what the resident was talking about. The hospital's geneticist, the resident said, could tell them more. It was just after 2 p.m. when Kathy called Joe, sobbing, her trademark Irish stoicism cracking. Joe knew immediately that whatever Kathy was about to say was dire. His wife was congenitally calm. It took a lot to frazzle her. In fact, in the 4 years they had been married, Joe had rarely seen her shed a tear. Now she was incoherent. He felt his pulse quicken as he pressed his ear to the receiver, straining to decipher her words through the sobs and din of the hospital. She was hysterical, and her fear filled him with panic. Cystic what? He asked. He had no idea what she was saying, but he seized on the one word that he understood: fatal. He rushed straight from his office and sped to the hospital where Kathy sat, her face flushed and tear-streaked. Joe held her until they were led into the office of a medical geneticist, where they learned Joey's fate. The geneticist, short with a goatee and dark hair, shook Joe's hand, but barely nodded at Kathy. He reviewed Joey's records and, without making eye contact with either parent, delivered the facts with speed and clinical precision. This is a genetic disease, the doctor said. Everyone carries 2 copies of the gene that, when mutated, causes cystic fibrosis. Each of you carries 1 normal and 1 mutated version of this gene, and you each gave your son a bad copy. He inherited this from both of you, adding, receiving 2 bad copies is lethal. The words sounded garbled and foreign to the O'Donnells, as if the conversation was happening underwater in a distant ocean. Inherited? Neither Joe nor Kathy could remember anyone in either family who had died young, and both of them were in great health. They'd never crossed the threshold of a hospital before Joey's birth. The geneticist continued with his rapid-fire, monotone primer. Children with this disease have salty skin. They produce thick mucus that clogs the airways and promotes severe bacterial infections that destroy the lungs and lead to death. The life expectancy for children with this disease is 5 years, but your son's lungs are badly damaged, and he probably won't live a year, he said. Then he paused. He probably won't get out of the hospital, and if he does, it won't be for long. Your son's death is going to be slow and painful, and you ought to know that. The message was clear. Their baby was dying, and further investment by Joe and Kathy would be pointless. The geneticist's assessment left no space for hope. So that is the sad but very intimate beginning of Breath from Salt. Next page. Next slide. Next slide. So after I had heard Joe and Kathy's story, I was absolutely hooked on writing a book. And I realized that I had to tell the story of the O'Donnells. And when the O'Donnells got Joey's diagnosis in 1974, they immediately turned to the Cystic Fibrosis Foundation. And this was a foundation launched by families who were particularly and personally affected by the disease. And this foundation began in 1955, about 17 years after Dorothy Anderson had first characterized the disease. Next slide. So in this picture here are Bob Dressing and Doris Tulson. And they were very forward-thinking members and leaders of this foundation. And they were the ones that realized that the only way to find a cure for this fatal rare disease was to raise money, because money buys science and science can yield cures. So Bob and Doris, two of these early influential leaders, they poured all their effort and initiative into raising money and engaging parents, friends, and family to do the same. And they also organized a panel of scientists who would review research proposal and grants so that the money was given to the most promising scientists. Next slide. So there is the story of the patients and the families, and then there is the story of the Cystic Fibrosis Foundation and what science they actually funded, because they believed that if they funded science, they would eventually find a cure. So the money that they raised was used to look into the gene and to cure the disease with gene therapy. Next slide. So this was the second element that really, the second thread of the story that went throughout the book. And the reason is that cystic fibrosis has had an oversized impact on science and medicine. And I wanted to tell the story of these scientific breakthroughs through the eyes of trailblazing scientists who made the discoveries. And I wanted to write this book. And as I wrote, I wanted to meet as many of these people as possible to learn how they did their experiments, why they studied this disease, and what motivated them. This was definitely one of the most fascinating and enjoyable parts of the reporting. Next slide. All right, so this is Dorothy Anderson, the woman I mentioned, who first discovered cystic fibrosis. Now, needless to say, she made her discovery in 1938. I did not interview her. But, you know, often today when we talk about cystic fibrosis, the discussion begins with this discovery of the gene, but the story begins much earlier. It was Dorothy Anderson, a largely forgotten character in the history of medicine, who actually characterized the disease back in 1938. And she was the first to recognize that CF was a distinct disease. And she noticed that it had been mischaracterized for celiac disease, which I'm sure most of you know is an allergy to gluten that can be cured essentially with a special gluten-free diet. And she was a truly extraordinary woman for her time. And I'd like to read you an excerpt from the book and tell you a little more about her. Cigarette hanging loosely from her lips, pathologist Dorothy Hansine Anderson peered through a veil of smoke at the little girl lying on the metal table in front of her, the barest warmth long absent from her translucent skin, a reminder of her untimely, unnatural death. In 1935, Dr. Anderson was a rarity in America. Fewer than 5% of practicing physicians were female, and they were strongly encouraged to focus on just pediatrics, obstetrics, gynecology, housekeeping elements of public health, and preventative medicine. And also social pressure at the time dissuaded patients from actually seeing female physicians. But Anderson was one of an even smaller group of women who held both medical and a doctoral degree. She was pragmatic and worked tirelessly, married only to her work. She shunned makeup and wore her hair in a tight Victorian bun. She chain smoked and, after hours, drank hard. As a pathologist, Anderson autopsied bodies of patients whose deaths were inexplicable or of academic interest. It was a profession suited to her solitary nature, and she took pride in the methodical work it required. Once patients' bodies were delivered by gurney to the hospital's dank bowels where Anderson presided, the highly choreographed autopsy procedure began. There was a specific order for weighing, measuring, cutting, and sectioning each organ. Anderson would take small slivers of suspicious tissues and send them to a histology lab, where the tissue would be dyed, embedded in paraffin wax, sliced paper thin, so she and others could examine it under a microscope. Now she had a new case, which she labeled MD, case 44. With the child lying on the table, Anderson opened her notebook, arranged her instruments, and began the autopsy. Age at death, 3 years and 1 month. The body was pale but well nourished. A bright red slash across her right clavicle was where surgeons had corrected her twisted neck. The contour of her chest was normal, with a plateau at the chest bone. The belly slightly distended, but greatly improved from a year ago when Dorothy Anderson saw her. With a scalpel in her hand, she scored the girl's chest and inserted a surgical buzzsaw, grinding her way through the sternum. She cracked the breastbone in half and stretched the ribs apart like 2 halves of a walnut. The heart and aorta, the hose-like vessel that carries blood to the rest of the body, looked normal. She removed the heart, weighed it, and put it aside. She removed the right lung and weighed it and filleted it, opening it like a book. As a pathologist, she'd seen many healthy lungs, spongy, soft, and pink, but this was not one. A healthy lung could be squeezed, chasing pockets of air into other chambers of the organ that then expanded balloon-like. This one was heavy and dense, pocked with dime-sized abscesses that were filled with plugs the color of dried blood or the hue of yellow cheese. Anderson knew that the color was dependent on the bacteria living there. The tissue between the abscesses was mottled and tough and incapable of the stretching and relaxing that breath inspired. The bronchial airways were cemented with thick, sticky mucus. The left lung was a little lighter than the right, but when split open, it looked exactly the same. Next, Anderson removed and weighed the soft, bright red spleen. It looked normal. The liver was pale yellow rather than reddish brown, but otherwise normal as well. The stomach small, but normal. She then lifted it to examine the pancreas nestled below. Something wasn't right. Anderson leaned in for a closer look and then removed the pancreas to place it on the scale. It was just half the expected size, and she inserted her scalpel into the banana pepper-shaped organ. She heard a scraping sound, as if the pancreas were filled with grit. The healthy pancreas is an unsung gland sitting discreetly in the abdomen, shielded by the liver and stomach, quietly secreting various juices. Some of its cells make insulin, a hormone vital for lowering sugar levels in the bloodstream, while others make a hormone called glucagon, which increases blood sugar when the body needs energy. Other cells in the pancreas churn out acidic digestive juices, without which the intestine cannot break down food and extract nutrients. But most of MD's pancreas, in contrast, was filled with fibrous cysts, something Anderson had never seen before, and had only a few remaining insulin-producing cells. In a healthy pancreas, a long central tube called the pancreatic duct runs along the length of the organ, collecting digestive fluids and passing them into the bile duct, which empty into the intestine. But when Anderson searched MD's pancreatic duct, she could find only a short half-inch section. The rest of the pipe, if it existed, was lost in a mass of scar tissue. It was clear that this organ couldn't possibly have been doing its job. That explained the patient's malnutrition. But Anderson had never seen a celiac patient with a pancreas that was so damaged. She began combing through the medical journals for clues, spending all of her free time hunting through stacks of books and journals in nearby Columbia University's library for mention of similar cases. And she explored autopsy files on other children who had been diagnosed with celiac disease. She soon discovered reports from Boston to Europe to Australia describing similar fibrous pancreatic cysts in other children who had been classified as celiac patients. But she knew kids with celiac disease, once they were prescribed the right diet, didn't usually die. They grew quite normally. Perhaps MD didn't have celiac disease at all. In fact, based on the state of the cystic fibrotic pancreas, she was sure she didn't. Surrounded by an ocean of medical literature in Columbia's library, Anderson realized that this could be an entirely new disease, one that, unlike celiac, was incredibly deadly. So that tells you a little bit, a peek into the life of Dorothy Anderson and the autopsy in which she discovered cystic fibrosis. Next slide, please. So here is just a quick diagram of what it was that she saw. Here you see the pancreas, you see a duct running through it, and that collects all the juices and delivers them to the intestine. In a CF pancreas, that duct has been completely destroyed. Okay, next slide, please. So Dorothy Anderson was also the first person who really got a sense of how the disease was transmitted from one generation to another. And even though this was long before the discovery of DNA and long before the discovery of a gene, what they did know is that both parents had to be a carrier of some sort of mutation, and that you need 2 copies of the mutation to get the disease. And so you can see that if the mother and father both carry a bad copy of the CF gene, then there is a 1 in 4 chance that the offspring will get that disease. Next slide. And so what's amazing is that although Dorothy Anderson really saw that the lungs were messed up and that the pancreas was deformed and not functioning, cystic fibrosis patients suffer from a full body disease. They have sinus problems, nose polyps, frequent infections in the lungs, which destroys the lung tissue and makes it very difficult to breathe. There's abnormal pancreas function, which, as I mentioned, that affects the digestion of food. And as a result, you get undigested food and fatty bowel movements. You also suffer from malnutrition. But probably the most unusual element of this disease was that all the patients that suffered from cystic fibrosis had hyper salty sweat. So what was the cause, and what could bring all these different symptoms? What could explain the cause of all these different symptoms? Next slide. Well, unlike Dorothy Anderson, whom I had to learn about by digging through the archives of Columbia University, I actually had the pleasure of meeting Paul Quinton, a scientist who has CF. This is Paul here. He and another team on the East Coast figured out the defect in the cell that causes his sweat to be super salty and triggers all the varied symptoms of cystic fibrosis. And my conversation with Paul was riveting. He explained to me what it was like to live with this disease in the 1940s and 50s, when very few physicians even had heard of cystic fibrosis. And it was even more thrilling to learn how he diagnosed himself and then spent the next 20 years figuring out how his cells were misbehaving. In fact, Paul gouged out chunks of his own flesh to figure out why his sweat glands secreted saltier sweat than other people with this disease. Next slide. So here's a close-up of a sweat gland. So on the left, you will see the coil of the sweat gland and a droplet of sweat on top. So in normal sweat, you excrete all this salts, so sodium and chloride, both the 2 halves of the salt molecule. And your cells that line the sweat gland snatch all this salt back into your body so you don't lose all these electrolytes. But with the cells in a cystic fibrosis patient, their cells are defective. They can't pull back all that sodium, all that chloride. And so as a result, they lose their electrolytes and they suffer from very, very salty sweat. And when you can't regulate salt and water in your body, it doesn't just affect your sweat. When the salt water balance is off in the lungs, the result is very sticky mucus that allows bacteria to live and destroy the lung tissue. Okay, so picking it up a bit. Next slide. So the Cystic Fibrosis Foundation, once they found out what the cause was, they realized that they had to discover the source of the problem. Next slide. And they began to fund the hunt for the gene. And this is Lapchi Choi. He used gene hunting techniques to find the cystic fibrosis gene on chromosome 7. Next slide. And then he teamed up with Francis Collins' group at the University of Michigan, and they found the whole gene. And they found out that this was a gene that was involved with balancing salt and water. Next slide. Now, immediately, people thought that, okay, if we have the gene, we can use gene therapy to fix this disease. But that wasn't the case. It wasn't easy to give a sick person a copy of a healthy gene and put it throughout their body. But one of the upshots that was so great was that the O'Donnells were able to use gene testing, prenatal screening, to have healthy children after they lost Joey. And as you can see in this picture, we have Joe and Kathy on the right, their daughters, Casey and Kate, and now Casey has her own family too. So that was an immediate upside of discovering the gene. Next slide. Now they tried to put the healthy gene back into a sick patient. And while the theory worked in principle, it had been tested in the lab, it had been tested in animals, and it sort of worked in humans, but getting a healthy gene into the lungs of a patient is very, very difficult, and they couldn't get a therapeutic effect. And this is where the arc of the story changes. Next slide. So genetic testing was great. Families could now screen to see if they had a bad CF gene, but gene therapy was a failure and they didn't get a cure. Next slide. So this was when they realized the CF Foundation realized they had to adopt a completely different strategy. And so what they did was they used venture philanthropy. Now, I've never heard of venture philanthropy before I started writing this book. And basically it involves raising money through philanthropic donations and using this money to fund the development of drugs directly from biotech and pharmaceutical companies. And even though Joe lost his son to cystic fibrosis, he's a splendid businessman and has raised billions for Harvard University. So he directed his fundraising abilities for the Cystic Fibrosis Foundation. He raised tens of millions. And then Bob Bell, who you see at the top here, and Preston Campbell, these two leaders of the Cystic Fibrosis Foundation, they found a biotechnology company and they charged them with funding the early R&D. And they said, we want you to develop a drug that can fix the broken protein in CF patients. Next slide. So they gave the money to this company called Vertex Pharmaceuticals, and Vertex thought, yes, it's never been done before. We've never been able to, nobody on earth has been able to fix a protein that is broken in cells and make it work normally. And so they accepted the money from the Cystic Fibrosis Foundation. And in exchange, they said, all right, if we get a drug, we will give you royalty payments for that drug if it actually goes to market. Next page. And so here's what was actually wrong. This protein, which is this green glob at the top, it was broken. Now, in cystic fibrosis, there are more than 2,000 mutations that can cause cystic fibrosis. So Vertex chose to work on the simplest one just to see if it would work. So this protein is stuck shut. And I call this the door jam mutation because basically it won't let any chloride out. And so the foundation said, all right, Vertex, can you find a molecule that will open up this protein and let chloride flow normally? Next slide. And indeed, that's exactly what they did. It took many years, but they found a molecule that could open this cystic fibrosis protein that was broken, and they could force it open and chloride would flow normally after that. And when that happened, the patient's lungs began to empty out all the mucus that had been clogging them and making them sick. They were able to cough it out, and many of the problems and symptoms of cystic fibrosis ceased to happen. So that was the first drug. Next slide. And this is a family, the Cheevers family. Both daughters have cystic fibrosis. They took the drug Kalydeco, and now they are in college. One of them has finished college. They no longer get sick. They don't get cleanouts at the hospital. They've been able to put on weight, and that's something they struggled with before. And now their life expectancy is normal. It's in the 70s and 80s like the rest of us, and they're going to be able to go on and have a family of their own. Next slide. So over the last 8 years, Vertex has been able to generate 4 drugs, including Kalydeco, that actually work for patients, for 90% of patients with cystic fibrosis. And as a result, Vertex made a huge amount of money, and the foundation got royalty payments. And now they are using those royalty payments to develop treatments for the remaining 10% of patients who don't have a cure or a treatment. Last slide. So I wanted to tell you, after telling you this story in light speed, I have great hope, not just that the cystic fibrosis community will find treatments for the remaining 10% of patients who can't use the drugs from Vertex, but that this their strategy will form a roadmap for other communities and other people with genetic diseases. And I think this is something that others can follow and hopefully make progress in curing their own diseases. So thank you so much for having me and having me speak here, and open it up to a few minutes of questions if we have that left.
Thank you so much, Bijal. I really appreciate the wonderful overview. I got a chance to read this book this summer, and it was wonderful to hear the shortened version of it today. But thank you for highlighting everything. We do have a couple questions. We're limited on time, so we might only be able to take 1 or 2. But let me go ahead and just ask this first one here. First, they have a comment. Thank you for this presentation and for reading from your awesome book. I also wanted to say that you helped me better understand the disease and cleared up misconceptions of it. I knew someone who died of it, but thought it was a lung disease, so thank you. Also, we have another question here of what is the next book you might be working on?
Well, right now I am a science editor at National Geographic, and all my energy is taken up covering COVID, which is something that the Cystic Fibrosis Foundation has been very concerned with, naturally, because that's a lung disease, and so is COVID-19. But I do have a story that I want to tell, but not quite ready to go public with it.
Well, we will look forward to hearing it when you if you are ready to go public with it. So thank you for that. I don't see any other questions at the moment, so I think that will wrap us up for this session. And I really appreciate, again, your time and your dedication to the cystic fibrosis history and story.
Thank you. Thanks for having me, and stay well, everybody.